Welcome to the deep dive. Today we're going straight to the very foundations of life, tackling a mystery that has puzzled evolutionary biologists for decades.
It really has. We're talking about the origin of the eukaryotic cell.
Right, the complex cell, the kind that makes up you, me, trees, fungi.
Basically anything with a nucleus and all those specialized internal compartments.
And our mission for this deep dive is to try and settle a huge timing question. Did the host cell that eventually became us get all its beautiful, complex machinery after it acquired its tiny power-generating partner, the mitochondrion? Or was that host already, highly complex before the merger?
This is the classic mitochondrion-early versus mitochondrion-late debate. And for years, the big challenge has been that evolution, well, it didn't leave behind a nice, neat trail of breadcrumbs.
No fossils of the in-between steps.
Exactly. We have no living, awkward teen versions of the earliest eukaryotes to study.
So we need a time machine. And our source material for this dive gives us one of the most brilliant evolutionary tools there is. It's called dated gene duplications. For listeners who maybe haven't tracked this before, how exactly does a gene duplication act like a molecular timestamp? How does it show us the maximum age of a new feature?
It's a fantastic concept. It's essentially a relaxed molecular clock. So think of it like this. An ancient organism has a single gene, and that gene does one job.
Okay.
But if that gene accidentally gets copied, duplicated, you now have two versions. We call them paralogs. And over time, they can evolve to take on two distinct specialized roles.
Like one for structure and one for movement, maybe?
Precisely. And the date of that original duplication event, that split, gives us the maximum age for when that new specialized function first could have arisen in that lineage. That's our timestamp.
And that is the absolute cornerstone of this whole discussion. We're tracing when the molecular toolkit for complexity was first put together, and by comparing these timestamps, we can build a timeline that points to a new model. A model we need to really spend time with today. The complexified-archaeon late-mitochondrion model.
Or column, for short.
N-A. I like it.
The column model basically proposes that the host cell wasn't some simple blob waiting for an energy boost. It was already a structurally and genetically complex entity. It was ready with things like a cytoskeleton and even a protonucleus before it ever merged with that alpha proteobacterium.
The one that became the mitochondrion. Okay, let's unpack this. We need to frame the story with a few key time points that come from this massive molecular analysis.
We do. We're tracing this evolutionary merger between an ancient Asgard archaeal host and an alpha proteobacterial endosymbiont.
So we need three key dates.
3 dates. First, the starting line for our host's solo journey. We call this the Enfica, the nuclear first eukaryotic common ancestor.
And this is?
This is the moment the future eukaryote lineage branched away from its closest living archaeal cousins, which we now know are the Asgard archaea. And that point is way, way back.
How far?
3.05 to 2.79 billion years ago.
Yeah.
We're deep in the Mesoarchaean era.
Okay, so that's the start of this long, slow burn of evolution for the host. What's next?
Next comes the moment of the merger, the big event. We call this the Mefica, the mitochondrial first eukaryotic common ancestor.
This is when the endosymbiont comes on board.
Exactly, when the eukaryote lineage branches from its closest bacterial kin. And this happens significantly later, around 2.37 to 2.13 billion years ago.
And the final date, that's when we get to the ancestor of all of us.
That's the LSEA, the last eukaryotic common ancestor. This is when modern eukaryotes really radiated out and diversified. And that happened around 1.80 to 1.67 billion years ago.
So what's critical here is the timeline, the time between these events.
The timeline is everything. The Enfica stem, so the time the archaeal host was evolving all by itself,
The solo journey?
was roughly 1.1 billion years long.
Yeah.
But the Enfica stem, the time after the merger leading up to LSA, was only about 0.6 billion years.
So almost double the time for solo innovation compared to partnered innovation.
That's it. So the idea that complexity just exploded after the merger doesn't fit. The host had a billion-year head start before its energy partner even showed up.
Let's use that immense time gap. Let's dive into our first section. Establishing this complex archaean host. Where did the building of complexity actually begin?
It began with the absolute basics. The structural components, the ability to move, to change shape, to divide. It all started with the cytoskeleton.
And eukaryotes inherited some of these components from their Asgard archaea ancestors, right?
They inherited the basic building blocks, yes, but they turned them into something vastly more useful.
Okay, let's start with actin. It's fundamental to cell shape and movement.
Our dating shows that the crucial actin gene duplications really kicked off around 2.8 billion years ago. Now, here's where the specific function really matters.
Go ahead.
We see very early duplications of two genes called ARP2 and ARP3. Their duplications date from around 2.9 to 2.1 billion years ago.
And why are those two so important?
This is huge because in modern cells, the ARP23 complex is the molecular machine you absolutely need to form branched actin filaments.
Okay, let's simplify that. If standard actin is like, I don't know, a straight piece of rebar,
Good analogy.
then branched actin is more like a grappling hook. Or maybe a net.
A net is a perfect way to put it. Because what does a cell do with a net?
It catches things. It eats.
It eats.
Yeah.
Branched actin structures are essential for phagocytosis, the ability to engulf large particles, including other cells or bacteria. Our data shows the capacity for phagocytosis existed well before the mitochondrial acquisition, which we've dated to around 2.2.
So the host cell had already built the molecular machinery it needed to consume its environment.
Long before it got its powerhouse.
That just completely blows up the traditional idea that the cell needed this massive energy boost from the mitochondrion just to afford the physical mechanics of eating.
It does. It shows the physical capability was there first. And we see the exact same pattern with tubulin, the other core structural protein.
The one that forms microtubules.
Yes. Its big expansion happened between 2.8 and 2.2 billion years ago. And what's fascinating is what came first. The first key duplication was for something called gamma tubulin, around 2.8 billion years ago.
And gamma tubulin's job is?
Its job is to create nucleation sites. They're called microtubule organizing centers, or MTOCs.
I love the factory analogy from the source material here. If gamma tubulin is the architect, it's setting up the organizational blueprint. It's drawing the factory floor layout, defining where all the internal highways inside the cell should run.
Precisely. The cell built the organizing system first. The actual core building blocks that form the final microtubules, the alpha and beta paralogs, they emerged a bit later, but still largely before the mitochondrion arrives.
So the big picture here is that the physical infrastructure for directed movement, for organized cell division, was all in place by about 2.7 billion years ago.
That's right.
Okay, so if this complex archaeal host was structurally organized, it was capable of cell division, and it was equipped with the molecular tools to actively grab and manipulate its surroundings, all a billion years before it got its full-time power source. Then we have to ask you, the listener, how fundamentally different was this complexified-archaeon from the simple passive archaea that we tend to think of today?
It was a different beast entirely.
All right, let's do a quick recap of this first phase. It's about 5 minutes in, good time to pause.
Good idea.
We've established this long, patient assembly process. It's not a single event. The starting gun fires with the Enfeka divergence way back around 3 billion years ago.
And then we see the cytoskeleton expanding between 2.8 and 2.2 billion years ago. Both actin and tubulin systems are getting more complex.
And the key implication, the real takeaway from this part, is that the molecular capacity for phagocytosis, the ability to eat, was operational before the mitochondrion even entered the picture.
The host was already a predator.
Okay, so we've established the skeleton. Now, what about the guts and the brain of the cell? Let's move inside and talk about that second wave of complexification. Building the internal infrastructure.
Yes, the endomembrane system.
Which is the system of all those internal compartments, right? The ER, the Golgi, the lysosomes.
Exactly. The systems for synthesis, for trafficking and shipping, and for digestion and recycling. And our dating tells a really coherent story about the sequence of events.
Which is that?
The components for making and moving things were elaborated before the components for digestion.
So you set up the factory's assembly line before you hire the cleanup crew.
That's a perfect analogy. The earliest duplications, between 3.0 and 2.3 billion years ago, involved proteins for trafficking. We don't need to get into all the names like snares and rabs.
Yeah, let's not.
But think of them as the cell's internal post office and its fleet of delivery trucks. These proteins manage budding, fusion, and transport between the ER, the Golgi, and the cell surface.
It makes sense. You build the fundamental shipping and receiving infrastructure first.
You have to. Then, the later duplications, which really start picking up around 2.4 billion years ago, they focus on specialized digestion, specifically the endolysosomal system components.
The endosomes and lysosomes. The recycling centers.
Right, and this timeline is just incredibly coherent. First, the host developed the capacity to physically grab things with phagocytosis, around 2.7 jacked.
And then, 300 million years later, it really elaborates the internal machinery to process and digest whatever it grabbed. Was this all built from the host's own archaeal genes? Or was there some bacterial influence already creeping in?
That's a great question. The initial framework, the genes for the ER-localized lipid pathways, those were archaeal-derived. They duplicated early. But yes, we do see key bacterial genes involved in lipid biogenesis duplicating later, maybe around 2.5 Eukaryotic. This suggests the host was already engaging in some horizontal gene transfer. It was borrowing genes and integrating new metabolic pathways, likely from non-mitochondrial bacteria.
So it was already tinkering with its membranes, maybe preparing for that final great merger.
Certainly looks that way.
Okay, let's move to the control center, the nucleus. This is a tricky one because the nuclear pore complex, that gatekeeper of the nucleus, is a pure eukaryote invention. We can't trace its ancestry back directly.
We can't. So instead, we have to look at the specialized jobs the nucleus performs. And one of the most important is RNA processing.
Specifically splicing.
Splicing. So we examine the molecular machinery for that, the little scissors and glue, specifically the SUSM and LSM protein complexes.
And these are essential for editing the raw RNA transcript before it can be used to make a protein.
They are. And their duplications are dated broadly from about 2.9 to 1.8 billion years ago.
That's a huge span of time, but the early edge of that is still well before the mitochondrion.
It is. But the real kicker here is the synchronous duplication of the various RNA polymerases. Pol first, Pol 2, and Pol third.
The machines that read the DNA.
Right. And Pol third, for example, is responsible for transcribing a key part of the splicing machinery itself. And its core components diverged around 2.73 to 2.32 billion years ago.
And the other polymerases duplicated around the same time.
They did. This strongly implies that the host was already separating the job of reading the DNA into specialized teams. It was a highly organized process, and it was happening before the MFECA date of 2.2 Ga.
And here's where it gets really interesting for me. This kind of sophisticated gene regulation, like splicing, it benefits hugely from being physically separated from the protein-making machinery.
It almost requires it. You don't want to start editing the instruction manual while it's actively being printed and used on the factory floor.
Chaos. The nucleus provides that separate editing room.
Exactly. And this early timing 2.9 to 2.3 gas, strong molecular evidence that the prerequisites for that spatial separation for a protonucleus were already being established.
So let's do our second recap. We're about 10 minutes in. Between 3.0 and 2.1 billion years ago, the archaeal host was building its internal world.
The post office systems for trafficking were set up first. Then came the digestive systems, and all the while, the editing teams for sophisticated gene regulation were already becoming specialized.
So by 2.2 billion years ago, this was functionally a complexified-archaeon.
It absolutely was.
Which raises another important question for you, the listener, to chew on. If the molecular machinery for splicing, which works best in the isolation of a nucleus, was already present, what non-energetic selective pressure could have been driving the nucleus to form before the huge energetic boost of the mitochondrion?
It's a fantastic question.
And now we get to it. The culmination of this billion-year buildup, the acquisition of the power plant.
The main event. We can align mitochondrial endosymbiosis, the MPECA, precisely with the onset of alpha-proteobacterial gene duplications. And that happens at roughly 2.2 billion years ago.
And the data shows an immediate acceleration in evolution.
An explosion. Duplication rates just spiked shortly after MPECA. The host went from this slow, steady, patient innovation to hyperspeed.
And this is the moment where endosymbiotic gene transfer, EGT, really kicks off.
Yes, genes from the alpha-proteobacterium start jumping ship into the host nucleus in massive numbers.
And what's the direct evidence of that immediate genomic reorganization?
We can see that some of the very earliest alpha-proteobacterial duplications include genes whose second copy, their paralog, immediately took on a non-mitochondrial role.
So they were repurposed instantly.
Instantly. For example, an RNA methylase called METTL4, it duplicated right around 2.2 JAG, and one copy now functions exclusively in the host nucleus. The genes were acquired and immediately plug into the host's existing complex infrastructure. It's the smoking gun for the start of integration.
And we can also date the creation of the physical infrastructure needed to manage this new partner, can't we?
We can. We can date the mitochondrial pre-protein import system. These are the channels that let the host import its own proteins back into the mitochondrion, which is essential for control.
Right.
Duplications of components like TM14 and TM44 happen between 2.24 and 1.99 billion years ago. It provides a maximum age for when that host-symbiont interface was first established. The timeline is incredibly tight, The merger happens, and the machinery to control it is built right away.
It's often really tempting to tie this timing to the major planetary shift happening around then, the Great Oxidation Event, the GOE.
Right, when atmospheric oxygen levels shot up.
The mitochondrion is aerobic. It loves oxygen. So doesn't that just make perfect sense?
It makes sense as important context, but not as the sole driver. Because remember, the entire formative stage of our complexified-archaeon, from 3.0 to 2.2 Syrah, that all happened under anoxic or, at best, hypoxic conditions.
Low oxygen.
Very low oxygen. So if the host was already complex and ready to eat before the GOE, then the GOE might have provided the necessary environmental pressure for this specific partnership to stick. But the anomplex structure itself was built in a low-oxygen world.
Which supports some of those other hypotheses.
It supports syntrophy hypotheses, which suggests the original driver was some kind of mutual metabolic dependence, maybe involving hydrogen or sulfur, rather than just an urgent need for an oxygen-burning power plant.
Okay, so we can use these really firm time constraints now to definitively test some of the competing origin stories. Let's start with the mitochondrion-early model, like the famous hydrogen hypothesis. Why were those so popular for so long?
Because the energy argument was just so compelling. You know, big, complex cells need a ton of energy. It's a real constraint. So they theorized that the energy problem had to be solved first.
But our data refutes that.
It refutes it directly. All this complexity, the cytoskeleton, the nucleus prerequisites, was demonstrably present before the merger. The host wasn't energized into complexity by the symbiont. It was already highly capable.
Okay, what about the serial endosymbiosis model? The idea that the host sequentially gobbled up multiple different bacterial partners before the mitochondrion.
We can test that too. We analyzed duplications of genes from non-alpha proteobacterial sources. And the vast majority of those duplications that happened before Lesia, they largely post-date the 2.2 billion-year mitochondrial acquisition.
So the other bacteria were acquired later.
Yes. It suggests the events didn't happen sequentially beforehand. Rather, acquiring the mitochondrion seems to have supercharged the host's ability to acquire and integrate genes from other bacteria later on.
Which brings us right back to the CAM model, the complexified-archaeon late mitochondrion.
Exactly. The archaeohost spends a billion years assembling its own architecture. The cytoskeleton, the ability to eat, the foundations for the nucleus.
Then, around 2.2 billion years ago, it acquires the mitochondrion. And with that new power supply, it rapidly diversifies into the last eukaryotic common ancestor by about 1.8 Jack.
The structure was the prerequisite, not the consequence of the energy.
So what's the core takeaway here? The big picture.
I think the core takeaway is that this research moves the discussion beyond just, plausible stories.
Yeah.
It establishes a precise, constrained sequence of events. And the key insight is this. Complexity, the internal architecture, the molecular framework, that was the foundation. It was the prerequisite for mitochondrial acquisition.
Not the result of it.
Not the result. The host was already a highly sophisticated cell, just waiting for the right energy source to finalize its structure and take over the world.
That really does flip the decades-old narrative on its head. We found that something like 85% of the crucial archaeal-derived gene duplications, the actual structural tools, were fixed before the mitochondrion ever arrived.
The blueprint was already there.
Which suggests the evolution of complex life wasn't just an energy problem solved by a lucky encounter. It was an internal architectural project, driven by the host's own ability to innovate for a solid billion years. So we'll leave you, our listener, with this final thought. If the energy boost from the mitochondrion wasn't the trigger needed to start the building process, what fundamental capability did this complexified-archaeon possess? Maybe an unprecedented ability to accumulate and manage huge numbers of duplicate genes, or maybe develop systems to handle a sprawling, messy genome. What allowed it to complexify for a billion years before finally sealing the deal with its powerhouse? Something to chew on during your next deep dive.
Until next time, stay curious.